Hi everyone. So in the last video, we talked about DNA replication and the structure of RNA. So now let's go on to transcription, which is how DNA will create RNA, and then translation, which is how RNA can then produce a protein. So transcription will be pretty short here. Basically what happens is our DNA double helix is going to unwind itself at a particular spot. And there's an initiation point and a termination point. Then nucleotides are going to come in to create messenger RNA. So the messenger RNA is going to be a complementary strand to our strand of DNA. So you can see here we've created an mRNA strand just based off of the information in our DNA strand. And remember, when we're creating a complementary strand, it goes opposite to the original strand. So if our original strand had 3' at the top and 5' at the bottom, our complementary strand will have 3' at the bottom, 5' at the top. All right, so once we create this strand of RNA nucleotides, then it separates to become its own strand that can then go on to say, hey, I'm ready to make a protein. DNA will just rewind itself and then potentially make more RNA. So this whole reaction is catalyzed by an enzyme called RNA polymerase. So RNA is what we're making, and then we're creating a polymer, and it's an enzyme, so it ends in ASE. All right, so that was pretty short and easy. So now let's look at the process of translation. So how do we go from RNA to a protein? So the messenger RNA that we just made is going to serve as a template on which amino acids are assembled in a proper sequence to make a specified protein. So remember, the primary structure of a protein is the linear sequence of amino acids. So the messenger RNA has basically an embedded code in it that's going to tell transfer RNA which amino acids to grab and which order to put them in. So an exact word-to-word translation is made from mRNA to tRNA, so messenger to transfer. Now this translation is called the genetic code, and it's considered a code because messenger RNA is going to contain 3-letter blocks that can be translated into amino acids, and these are called codons. So for example, maybe we have CGC, ATG, just some 3-letter block, and these are going to be specific nucleotides, right? Oh, except we don't use T in messenger RNA. Oh my goodness, what am I thinking? That's U. We don't have thymine in RNA, right? So we use uracil instead. So these are nucleotide bases, and these are the codes that are going to tell us which amino acid to bring in, and that's transfer RNA's job. So for example, in your textbook, there's a table that has the mRNA codons for each of the 20 amino acids, and some of them have multiple codons. So for example, let's look at alanine. If we need to put alanine into a specific spot to synthesize a protein, then messenger RNA will either have GCA all next to each other, GCC next to each other, GCG, or GCU. So all of these will tell us, or tRNA actually, tRNA will know, I need to bring in alanine. And then some of these amino acids only have maybe a couple of codes. So cysteine, for example, has UGC and UGU, and both of those will translate as cysteine. Now there's another table in your textbook that kind of goes over general characteristics of the genetic code. So again, codons are three-letter words. So alanine, for example, could be GCA. And the code is degenerate. So while GCA equals alanine, there's also three other codes that equal alanine as well. However, the code can also be very precise. So for instance, GCC represents only alanine and not any other amino acid, which is a good thing because remember the sequence of amino acids is very important. Because if we're a few amino acids off, that could change the whole function of the protein. Now there is a code that's used, or a codon that's used to say, hey, start here. The starting point is over here, and that's AUG. And then there's also three different codons that will tell mRNA or tRNA to stop. So once you're done synthesizing a protein, you'll see either UAA, UAG, or UGA. So those all just mean stop, we're done, we made our protein. Okay. Now this code is also almost universal. So for instance, in all organisms, pretty much, alanine has the sequence GCA for its codon. So that this is a code that's pretty true across all organisms. All right, so let's go through the steps of translation. So this is how we actually make a protein. We're finally there. Okay, so in step one, we have chain initiation. So what happens is messenger RNA is going to join up with a small ribosomal subunit. Remember earlier we said that ribosomal RNA is present in ribosomes. Ribosomes are the site of protein synthesis. So let's just kind of write that down to remind ourselves. Ribosomes are the site of protein synthesis. Okay, so we've got our small ribosomal subunit, and it's connected to messenger RNA. And we need to find that initial sequence that says, hey, we're ready to start synthesizing a protein. So remember that was AUG. So the initiating codon, AUG, is going to align with what's called the P site of our ribosome subunit. So P stands for peptidyl. So we're going to start creating a peptide, right? We're creating a protein. So P is for peptidyl. Okay, so now that we've started our process, a specific tRNA is going to come in with methionine or something called N-formal methionine, excuse me. So we just call that FMET for short. And its code is UAC, so that perfectly aligns with AUG. So this is what really gets our protein started. Okay, so once we've started this whole process, then our larger ribosomal subunit is going to come in and attach to our mRNA and our small ribosomal unit. So basically, all of this has just signaled, hey, ribosome, we're ready to start. We're ready to make a protein. Okay, so chain initiation, we've started the process. Okay, so now step 2, chain elongation. So now we're going to create our protein. And we're specifically creating that linear sequence of amino acids. All right, so we already talked about the P site. That's where we're initially going to start, but that P site actually is going to start moving down the chain. So this is where we're creating peptide bonds. Okay, so our P site has moved because notice that AUG, the initiation sequence, has now kind of shifted. So our next three letters are U, U, U. So that's messenger RNA saying, hey, I need the amino acid phenylalanine. So now transfer RNA is going to grab phenylalanine and come in and attach it to that F met that we talked about. So we're creating a peptide bond. Now this peptide bond is formed and catalyzed by peptidyl transferase. So that's another enzyme that's important here. Now, the next transfer RNA is going to come in and bond at a site called the A site. All right. Now, the A site, A stands for I can't say that word. Aminoacyl. There we go. That's a hard word to say. Aminoacyl. Okay, so let's say our next three letters are GUC. So that's going to translate as valine. So this is mRNA saying, hey, I need valine next. So then another transfer RNA will come in with valine attached. Now, they haven't connected yet. Phenylalanine and valine haven't connected yet. So the enzyme peptidyl transferase is going to come in and catalyze the formation of a peptide bond between phenylalanine and valine. So now we've created that bond. And notice that F-met is now no longer attached to our previous tRNA. So now we've got an empty tRNA, and that's going to just head on out. So we've got that empty tRNA just kind of going off to grab another phenylalanine if it's needed. All right, and then we just keep that process going. So the ribosome unit is going to move down the chain again. So notice that AUG is all the way over here. UUU has already been used up, so that's moved away. So now we're going to focus on the next three letters, which are AGU. And that's going to signal a tRNA to grab serine. Then peptidyl transferase will come in, create a peptide bond between valine and serine, and then we'll just keep moving on down the chain and just do this over and over again. Okay, so step 2 was chain elongation. Now, step 3, chain termination. How do we know when to stop? So remember, there were 3 stop codons that would tell basically this entire unit to stop because the protein has been synthesized. So just to kind of summarize here, we've got our 2 ribosomal units. They're going to come in and attach to messenger RNA at the initiation codon, which was AUG. Then the ribosomes are just going to kind of move their way down the chain. And as they go, they're creating peptide bonds between amino acids. So once this chain gets long enough, then we're going to start to see the secondary structure of our protein form. So remember, the secondary structure was either an alpha helix or a beta sheet. So that's going to start to form. Eventually, you're going to see tertiary structures forming as well. But at some point, the protein will be completely synthesized. So the messenger RNA will say, hey, stop. And it'll have that 3-letter code that it tells everyone to stop. So at that point, the ribosome subunits are released, we have our complete protein, and we're done. Okay, so let's review. Let's summarize the 3 steps of translation, and then we'll also list the steps involved in synthesizing a protein. So this is that flow chart that we've gone over multiple times. So go back, look at your notes, pause the video, make sure you know all these steps and kind of what they do, and then we'll go over it together. All right, so let's summarize translation. Okay, so the first step of translation was chain initiation. Okay, so this was where we had the initiating codon, AUG. So AUG is going to initiate synthesis. So we'll have large and small ribosomes, or ribosome subunits, I should say, that attach to mRNA. Okay, the next step, step 2, that is chain elongation. So this is where transfer RNA is going to start bringing in amino acids based on those 3-letter codons. Oh, and then the last step, step 3, I'll just put it over here because I ran out of room. Step 3 is chain termination. All right, and remember, chain termination is where we see a stop codon, and that stop codon is going to tell the ribosome subunits to detach from mRNA, and then our completed protein will also detach and go do whatever its function is. So whether it's helping transfer molecules through our body or transport them, or maybe they're muscle proteins, whatever their purpose is, they'll go off and do that. Okay, so now let's list the steps involved in synthesizing a protein. So this is that flow chart we've been looking at. Okay, so we said that DNA is going to turn into RNA, or create RNA, I should say. And what's the process there? Transcription. All right, and then RNA is going to create a protein, and what is that process called? It's right up above, right? Translation. So I used to get transcription and translation mixed up because they both start with T, so it's okay if you get them mixed up, but as long as you know how DNA creates RNA and then how RNA creates a protein, that's the important stuff. Also, in this flowchart, we said that DNA can replicate itself. So, if we have a new cell forming, we still need DNA for that cell, so DNA can replicate itself. Okay. Now, one last thing before we wrap up this chapter on protein synthesis. Remember that mutations are possible. So mutations are any changes resulting in an incorrect base sequence on DNA. Now, this could, down the road, lead to a mutation in a protein, right? So if you don't have the correct amino acid sequence, that's probably because you didn't have the correct codons in messenger RNA. Now, the reason you wouldn't have correct codons in messenger RNA is because DNA didn't have the correct nucleotide base sequence. So it all stems back to a mutation in DNA. Now, of course, issues could probably occur during transcription and translation that have nothing to do with the original DNA, but in general, mutations are going to come from DNA. And these can occur naturally during DNA replication. Something could go wrong. And this could also be induced by environmental factors. So ionizing radiation like X-rays or UV rays could disrupt or create a mutation. And then mutagens, which are chemical agents. Now, these mutations might not necessarily be that bad in the long run because cells are pretty good at recognizing that something's wrong. Next time, we're going to start chapter 23, which is on carbohydrate metabolism. And this will be our last chapter for the quarter, so I will see you then.